Hey guys, welcome to 14.3 as we continue to talk about genetics. In this section, we are going to talk about some really cool things that give you perspective into why, dog furs sometimes look different from each other, how puppies can have furs that look different from the parents, as well as what happens when we come outside of Mendel's pea plants, because when he talked about pea plants, he's talking specifically about, one gene that's affecting a certain trait. But what happens when we have more than one gene that affects a certain trait, like height or skin color? And so it's going to be a very interesting chapter. Please follow along, take good notes. And if you have any questions, always feel free to email me. Okay, so when Mendel was using his pea plants, the main reason he used pea plants was because they're simple. They are easy to mate, they're easy to reproduce because they make a lot of offspring. They don't have a huge amount of variation, but they're easily to be controlled. And each character in the peas, like their color, their height, except for their pod shape, which is, round, wrinkled, they're determined by one gene. All right, so the color is determined by one gene, the height's determined by one gene, but that one gene has two alleles, right? So we talked about like capital A, little a, capital A, capital A, little a, because one is dominant and one is recessive. But not all of the heritable traits are as simple, especially when it comes to human beings and animals. And even though they are a little bit more complex as you get further along talking about things that are not pea plants, we can still apply the principles that Mendel came up with in regards to segregation and independent assortment of alleles. And we can take that and apply it to more complex patterns of inheritance that we see in more complex organisms. So extending Mendelian genetics for a single gene. So let's talk about pretty much how we are deviating from focusing on one gene. So inheritance of characters that are determined by one gene, like we talked about the color of the peas and the color of the flowers, from the Mendelian pattern or the simple rules that he came up with, they are going to deviate in these following situations. So in these situations, we can't really follow Mendel's simple pattern that he came up with. So one situation is when the alleles are not completely dominant or recessive. So what happens if they're both, you have two things that are going to show up in one organism. So you could have two colors showing up in an organism because both of those colors are dominant or both of them are recessive or so on and so forth. Or when a gene has more than two alleles. Or when a gene produces multiple phenotypes. So they look different physically in more than one way. So there's a blend of colors, a blend of, mixture of that goes into creating a organism that's not dependent on just one phenotype or one genotype. So alleles can show degrees of dominance and recessiveness in relation to each other. So there are a couple degrees of dominance that we're going to talk about that will kind of help you understand different ways that we have certain traits show up. So for example, complete dominance. This is when you have phenotypes of the heterozygote and dominant homozygote are identical. So for example, in the F1 offspring, like in the pea cross, they looked like one of the two parental varieties because one allele showed complete dominance over the other. So this is basically in complete dominance, you're saying that you have one allele like capital A that is, or say capital P and little p, right? That capital P for purple flowers is going to outweigh that little p because it's going to show that flower to be purple. So in this case, in your F1 offspring, the offspring in your F1 are going to look like one of the parents that you originally mated with, okay? So that's why it's complete dominance. You're showing complete dominance over the other allele or the other trait. And so when it says heterozygote and dominant homozygote, that means if you're dominant, so you have big P, it's going to be purple. Or if you have, for homozygote, if you have big P, it's going to be purple. Or if you have heterozygote, big P, little p, it's still going to be purple. So they're both going to have identical phenotype, especially in the F1 offspring. So they're going to look like one of the parents because one of the parents had to be purple. All right, incomplete dominance. So in some genes, neither allele is completely dominant. Interesting. And the F1 offspring are going to show the phenotype that's going to be between those of the parental varieties, all right? So in this case, we're talking about that you are kind of having a blend of the two parents in your offspring. So for example, if we look at red snapdragons and I mate them with white snapdragons, some of your F1 offspring are going to show to be pink snapdragons. And that's because they are showing incomplete dominance, okay? So you will see this, for example, in snapdragons and some other organisms as well. Codominance is when you have two dominant alleles affect the phenotype in separate, distinguishable ways. So this is when you have Rowan cattle that I referred to before. If you have like a brown cow with a white cow and you put them together, they could show codominance because it could be a brown cow with white spots or white cow with brown spots. Blood types are also an example of this, and I'll go into this a little bit deeper because we have different blood types. You have A, B, but you have A blood type, B blood type, but you also have AB blood type, which is a combination of both. So when you have that, they're showing both of the blood types. Okay, so let's talk about the relationship between dominance and phenotype. So we call an allele dominant because it is seen in the phenotype, not because it subdues the recessive allele. So basically, we're not saying that it's dominant because it powers over the recessive allele, but we're saying that it's dominant because that's what shows up in the physical appearance of the offspring. And when you have dominant and recessive allele in a heterozygote, okay, so like big P or little p, they actually coexist and they don't interact. So you're not going to have the interaction of purple and white, but they're able to coexist equally and fine. They're not going to have a fight to see which one's going to overpower. And the only reason that, the only way that it matters if a allele is dominant or recessive is when we're taking the genotype and converting it to a phenotype. So when we're looking at the genes and we're trying to figure out what it looks physically, which is a phenotype. Okay, so to study this relationship, we can use Mendel's character when he studied the round versus wrinkled pea shape, the pea seed shape, okay? So when we looked at the round pea, this was the dominant allele, right? So the round was dominant over the wrinkle. So this means that the dominant allele, which is the round, is going to code for an enzyme that is going to convert an unbranched form of starch to a branched form in the seed. So I'm going to say that again. Dominant allele, which is the round, is going to code for an enzyme that's going to convert an unbranched form of starch into a branched form in the seed. And this is what makes the round pea seed appear round, and that's how it's dominant. But for the wrinkled pea seed, you're going to have a recessive allele, which is the wrinkles, and that's going to code for a defective form, all right? It's going to code for a defective form of this enzyme, which is going to lead to an accumulation of unbranched starch, which causes the excess water that is in the seed to leave through the process of osmosis. And when the seed starts to dry up after the water has left, it's going to wrinkle, hence you have wrinkled pea seed. All right, so the round pea seed is going to have this enzyme that's going to convert unbranched starch to branched. Wrinkled pea seed is going to have an accumulation of the unbranched starch, which causes excess water to leave the seed. and that's why it wrinkles as it dries up. Now, when we have something like, if I have big R and little r, right? So if I have big R and little r, this is heterozygote for round. So the R is now dominant, so it's going to mean that the seed is going to be round, right? So even if I have one dominant allele, that is enough. You'll have still enough of the enzyme to convert the starch from unbranched to branch. So that's why whether you have big R, little r, or if you have big R, which is also round, you are going to still have the same phenotype, which is round seeds. So whether you have two big R's, so you have plenty of enzymes to convert unbranched to branched, or if you just have one R, you're still going to have enough enzymes to convert the starch from unbranched to branch. And that's why they both are able to show round seeds, which is, I think, really cool, that we can kind of see the background of why a seed is round or why a seed is wrinkled. Okay, for any character, the observed dominant or recessive relationship is going to depend on the level at which we examine the phenotype. So there are, in some cases, different levels, and depending on which level you look is going to have a different type of phenotype that could be shown. So for example, you might have heard of something called Tay-Sachs disease, all right? So Tay-Sachs disease is basically a disease that is inherited. Sadly, it is fatal. And what happens is when a child inherits this disease, the brain cells of that child cannot metabolize these lipids, these specific lipids, because they have this enzyme that doesn't work correctly, okay? So normally we have enzymes in our brain that are able to process certain lipids so that our brain cells are healthy and well. But in children who don't have this enzyme that's working correctly, they can't process or metabolize these lipids. And so these lipids starts to build up in the brain cells over time, and the child will start to have seizures, they'll start to go blind, and they will start to have degeneration of motor and mental performance, which causes them to sadly die within a few years of age. So they usually don't make it past their childhood. But the only way that a child can get Tay-Sachs disease is if they have two copies of this allele, and they have to be homozygous to get the disease, okay? So even though they might be heterozygous for the disease, the only way they can have it is if they're homozygous. If they are heterozygous for this disease, they won't even show symptoms, so they'll be completely fine. You can also be a carrier for this disease and not even know it. So let's look at the different levels that I was telling you, and depending on which one of these levels you look at is going to tell you the phenotype of the disease or whatever it is that you're studying. So if you have an organism level, right? Basically, when you look at this level, the Tay-Sachs is going to be recessive, okay? But if you look at the biochemical level, the phenotype, which in this case would be an activity level of the enzyme, that is, whether it's able to process the lipids or not, is going to be incompletely dominant, okay? And therefore the heterozygous individuals will not show any of the symptoms, okay? So if you look at this example, and when I said, the different parts of dominance, remember that incomplete dominance is when you have neither allele is completely dominant. And so you can show the phenotype between the two parental varieties, so like the snapdragons, right? You kind of show a mix of it. Therefore, it's going to be heterozygous, and you usually won't show symptoms if you are heterozygous. If you look at the molecular level, the alleles are going to be codominant, which means that they affect the phenotype in separate, distinguishable ways. So, for example, like if I have two, like the AB blood type, they can show up in one blood type. Alleles at these different levels depend on which level you observe the phenotype. Like I said before, depending on which level you look at depends on what kind of phenotype you will possess. Okay, dominant alleles are not necessarily more common in a population than recessive alleles. This means that there are some cases that the dominant allele is going to be less frequently seen, but the recessive alleles are going to be more frequently seen. So for example, 1 in 400 babies in America are born with extra fingers and toes. That's pretty crazy. And this condition is called polydactyl, which basically is a rare dominant allele, and it's found less frequently in the population than the recessive allele. So the recessive allele is what is telling you, hey, we're gonna have normal fingers and toes, amounts of fingers and toes. But if you have this dominant allele, which is less frequent, so more rare, you will have an extra finger or toe. Multiple alleles. So Mendel's P's, we studied, have 2 alleles. They have big A, big A, or little a, right? But most genes actually have more than 2 alleles that form. So for example, if we look at the blood groups, right, we have A, B, O. Those are the 3 main blood groups. But there are 4 phenotypes that we can get from those 3. We can get blood type A, blood type B, blood type AB, or blood type O. And those are going to be determined by the 3 alleles for an enzyme that is going to attach the A or B carbohydrates to the specific blood cells. And that's how you get blood type A, blood type B, or I, which is just you have neither. So if a person can have blood type A, B, AB, or O, what this means is that these letters are referring to the 2 types of carbohydrates, A and B, that can be found attached to your red blood cell on the surface of the molecules. So I'm going to show you a picture, but A has a is distinguished in a certain way depending on how it attaches to the surface molecules. B, and then AB is a combination of A and B, and then O has nothing, right? And that's why people say O is universal. So enzyme that codes for the blood type A has an allele that adds the A carbohydrate to that blood. And then there's an enzyme that codes by the B blood type that has that is able to add B onto that blood type, and then same thing with the little I. So depending on which blood type you have, that's how it's going to add the carbohydrate to that. That enzyme will add that carbohydrate to that. So here's my fun picture of the 4 different types of phenotypes of blood that you can have. So the first one I have is type A. You can see that it kind of has these little funky pointy triangles on it. Then you have blood type B that has these little circles on it. Then you have blood type AB, which has a combination of both. And then you have O, which has neither. Okay, so this is kind of give you a visual of what our blood types look like. And so you can see how AB is going to be a lot more specific in when you're donating blood because it needs to match it correctly. There's something called pleiotropy, which basically in Mendelian inheritance, we saw genes only affected one phenotype, right? So purple affected, you got purple and stuff like that. But most genes actually have multiple phenotypic effects, and this is called pleiotropy. For example, pleiotropy alleles are responsible for the multiple symptoms that you can have in diseases that are inherited, like cystic fibrosis and sickle cell diseases. So let's see how 2 or more genes can be involved in determining a particular phenotype. So there's something called epistasis, which means in Greek, standing upon. And basically, this is when the phenotypic expression of a gene at one locus, remember locus is a specific location of the gene on a chromosome. So how one gene at a specific locus can alter a second gene at a different locus. This is so cool when you look at it. And I'm gonna use the example of Labrador retrievers, right? Everybody loves puppies. So, for example, if I say that capital B if I say that capital B is going to give me black. And if I say lowercase b, is going to give me brown, I'm going to use this as my first gene. So my first gene on my locus, on my chromosome, is going to tell me whether the fur on the puppy is going to be black or brown. Black is being more dominant than brown, okay? Now, I have a second gene on my chromosome at a different locus that is going to determine if there is going to be a pigment in the fur. So this gene that I'm going to use with the letter E is going to tell me if the puppy is going to be black or brown. And if it's going to show the color of black or brown, I need to make sure that my E is dominant, right? So it's going to be a capital E. If I have a capital E on my second gene, it's going to tell me that it's going to show a pigment. So it's going to show either black or brown on the puppy. If I have a little e, it's recessive allele. So that means there's no pigment. So if I have little e, that means I'm not going to have any pigment in the dog, and the puppy is going to look yellow, right? Or golden or whatever. It's going to look yellow. And so you've seen yellow labs. So how this works is, just imagine a chromosome, and on the chromosome you have two different loci, right? You have one locus with one gene, which is going to tell me if the puppy is going to be black or brown depending on the genotype. So if I have big B or big B little b, the big B is going to be dominant, so it's going to be black. Then I have a second gene on a different locus, and that's going to have either big E, which means that I'm going to show one of the two colors, black or brown, depending on what the first locus's genotype is. And then I could have little big E, little e, or little e. If I have little e on the second locus, it doesn't matter what color is on my first gene. If I have little e, I'm not going to have any pigment shown, and that puppy is going to look golden, okay? So for example, if I say that my first gene, let's see if I have here. So if I say that in our first gene, I have big B, little b, that means it's going to be black. But my second gene shows little e, which is recessive, that means there's no pigment. The dog will be yellow, even though the first gene says it should be black, okay? So if I said, okay, my first gene says big B, little b, so it's going to be a black puppy, and my second gene has big E, or big E, little e, I'm showing dominance, that there is going to be a pigment shown on the puppy. It's going to refer back to what the first gene says the pigment should be, and since the first gene says that the pigment should be black, it will show as a black Labrador, okay? That is something that kind of just blew my mind and so cool when I learned this, and I hope you think it's cool too. Okay, polygenic inheritance. This is when you have multiple genes that are independently affect a single trait. So there's a couple of definitions. Quantitative characters, those that vary in the population along a continuum. When you have quantitative characters, it's going to tell you that there is a polygenic inheritance. What is a polygenic inheritance? It's when you have additive effect of two or more genes in a phenotype. So in other words, you have multiple genes that independently affect a single trait. So for example, you have more than 2 genes that go into determining your skin color. You have more than 2 genes that go into determining your height. Actually, you have more than 180 genes that affect your height, okay? So that's why sometimes you could have, your dad super tall, mom super short, you're somewhere in the middle, or you could be taller than your dad or shorter than your mom, or so there's multiple things that go into that. Same thing with your skin color. You could have a blend of your parents' skin color, or maybe more like your mom, more like your dad, and that's because there's so many genes that go into these phenotypes that you can express. If we talk about skin color, this is just an example, so just bear with me as I get through this. So if I have 3 genes and each represent a dark skin allele, so if I have A, B, C, all capital. And then I have an incompletely dominant in the other allele, which is little a, B, and C. And if we put this together, we can say that if I have a person with capital AA, capital BB, capital CC, this person is going to be very dark because they're showing that dominant part of those alleles. But if I have lowercase all through AA, BB, CC, they're going to look very light, right? But if I have capital A, capital B, little b, capital C, little c, they're going to show an intermediate shade somewhere in between the super dark and the super light. So if I cross capital A, capital B, capital C, little c, try saying that 100 times, with each other, I would get offspring that are mostly somewhere between somewhere in the intermediate phenotype. So they're not going to be too dark, but they're not going to be too light. And also you have to keep in mind that there are other factors that can influence your phenotype, like your skin. For example, if you're on vacation in the Bahamas, you are going to come back looking a little bit darker than your parents that stayed back home, right? So there are environmental factors that can cause some change in your phenotype. Nature and nurture. So another thing that kind of takes us away from Mendelian genetics is when the phenotype for a character depends on the environment, as well as what is the genotype, so what's in the genes. So for example, if I have a tree, right, if I have an oak tree, obviously it's an oak tree because both of its parents were oak trees, right? So it's going to have a genotype that says it is an oak tree. But the oak tree might have leaves that are not exactly the same size, right? Not every single leaf on the oak tree is going to be the same size. Not every single tree is going to have this, not every single leaf on the oak tree is going to be the same exact shape. Not every single leaf on the oak tree is going to have the same green color. And this is because even though the genes of the oak tree says it's an oak tree, so it's going to have a specific shape, color, whatever, all the leaves are going to be varied because they are exposed to wind, right? That might slice one of the leaves and make it look different, or when it grows back, it looks different, or the way that the sun hits the leaves. Maybe one side of the tree gets a lot more sunshine than the other, so the color green is going to look darker on one side and lighter on the other. And then rain also, how much water is it getting? Is there going to be more water on one side, another side? How does the rain affect the tree in general? Same thing with humans, right? Even though you might have maybe obesity in your family, or you don't have obesity in your family, right? Even though that's not genetic, or that's kind of a bad example, but if you have human nutrition, right, and you have siblings, you could look different from your siblings because maybe you're eating something different from your siblings that's causing you to grow a lot faster than the other ones, or you could be exercising more so that you're actually building more muscle than your other siblings, or you are outside more and so you're getting more exposure to the sun, so your skin's a little bit darker than your siblings. So there's, even though you and your siblings have kind of the same genetic makeup, you guys could look very different depending on what exposure you're getting. So phenotypic range is going to be very broad in these polygenic characteristics because you have more variation involved. And then there's also something called multifactorial, which basically you have many factors that are both genetic in your genes and environmental that is going to influence how you physically look as an individual, or how an organism physically looks. So an organism's phenotype includes its physical appearance, its internal anatomy and physiology and behavior. So that all is included in the phenotype, because even though you can't see your internal anatomy, it's still a physical thing, right? And then organism's phenotype also reflects its overall genotype and unique environmental history. So because of your genotype, that's how we get your phenotype, okay? So because your genes are made up, whatever your genes say, that's how you're going to look, along with a mixture of your environmental history, so where you're located. I have cousins who are located in India who may have darker skin than I do, or I might have, I in reality, I have cousins in India who are a lot lighter than I am, and that's because of my parents versus their parents, or whatever it is. But I know this is a long section, but very interesting, lots of information, so please make sure you're taking well notes, and I'll see you in the next video.